Welcome to the important tutorial section, that is drug design. So in this tutorial we are going to see how we are going to design a drug against particular disease. Drug design is nothing but an inventive process of finding new medications based on knowledge of biological target. It is otherwise known as rational drug design. Applications of computer in drug discovery is simply called as computer-aided drug design. There are generally two approaches are available for drug designing. One is known as target-based drug design. The other one is known as ligand-based drug designing. In the case of target-based drug designing, we start the drug discovery process from receptor. In the case of ligand-based drug designing process, we start the drug discovery process by using a ligand or chemical modeling. So this is what the major difference between the structure-based or target-based or ligand-based and chemical-based. In the present tutorial, we are going to see about how we are going to do a structure-based drug designing, that is target-based drug designing. So the target-based drug designing is going to contain is carried out by using different steps. For example, first we have to carry out the target identification, and then we have to go to target validation and selection of drugs and hits, docking and ADME toxicity prediction, and then you select a best lead. So this is how the steps are involved for doing structure-based drug design. The target identification is a step. It is nothing but identification of a target which plays a crucial role in a particular disease or condition has to be identified first, and then whether the identified target is correct or not is identified by the process of target validation step. And then the third step is selection of drugs against the target, and then another one is the selection of hits by using this QSAR studies, and then docking of drugs and hits with the target, which is achieved by docking process, and then prediction of ADME values and toxicity values for the drugs as well as the hits, and then with this predicted values we can select a best lead, and that is how we are going to carry out this drug designing process through in silico method. So the first step is target identification. So how we identify a target for a particular disease? It's simply by searching the journal databases like PubMed or Science Direct or SpinkerLink. And the second step, that is the target validation, and it is also achieved only by searching the journal databases and drug bank databases. By using these two tools we can validate the presence of a target for a particular disease or a condition. So now how we are going to validate and identify the targets by using this PubMed journal database. That's what we are going to see. So through this drug designing concept, we are going to identify the drugs for HIV. So you should first open Google, and then you type simply as pathogenesis of HIV, and you search the Google, and then it will give a list of the search informations. In this you can go to the articles. Say for example, here I selected the pathogenesis of human immunodeficiency virus infection article, and then it will go to this article page. It gives only abstract. If you want a full article of this particular link, you should click any one of this link. I had clicked this link, and it opens the files of this link, and if you see this link, it is found to be a good link which give almost every information about the pathogenesis of human deficiency virus. By studying this full paper, we identified that how this HIV infected a human, and how it is actually affect a particular cell, and what are the parts of this HIV is going to play a vital role in its infection, and all the things are given nicely in this article. So this article is highly selected by browsing the Google by using this PubMed. Okay, so we get this particular article, and through this article identified the target as HIV proteases. The HIV will have different components in itself, but the components which are main important components of this HIV is found to be the enzymes, proteases, integrases, and reverse transcriptases. Of this, I selected this protease as a target for our drug designing project because there are so many drugs are available for reverse transcriptase and integrase. Only less number of drugs are available for proteases. That is why here we are selecting this protease as our present target. So this is how we are identified the target by using this PubChem research, and then you can keep on going to the next page of the search tool. Here also you get one more informative link is available here, which will give information about even how the HIV pathogenesis occurs. Like this, we have so many links which will give much informations about the HIV infection. See here, you will say the dynamic of genetic variants, how the mutation is going to occur in the HIV. So we have to identify, that is, we selected the target as a HIV proteases, and we have to identify whether this protease undergoes any mutations, any change in this. If HIV allows more mutation in this particular region, then you may not choose this particular gene as your target, because if HIV keep on changes the protein sequence of proteases, what is the use of using it as a target? So you have to validate your target again by searching the databases, and it this file is actually indicates that mutation mostly occurs in the polymerases part of the virus, not in the case of proteases and other things. That is how you can validate your target. So first we identify the target by analyzing the articles available in the PubMed, and second one, we validate whether our target is correct one, whether it is not going to undergo any changes like this by using this article, which clearly indicates that most of the mutations are occurs in the polymerases sequences, not in the case of proteases. So by studying these things, we have validated our target, that is proteases. So we identified our target as well as validated it. If you need the PDF format of this file, then you can click this link and then you will get the PDF format of your file. Once the PDF format is loaded here, you can save this PDF format by using the link save as options. Similarly, here is also it is loading. So once loaded, you have to use this save as option to save the file in a PDF format, which may be used for the future works. So by using this PubMed, we identified the target as proteases for the present study, and then we searched the PubMed databases and we validated our target. The next one is that we identified target and validated the target. Now you should get the drugs for this particular target. Then only we can able to carry out this QSAR study. So for that purpose, now we go for the databases like Drug Bank or again this PubMed journal databases to get information about the drugs available against HIV. So for this, you just add the word AIDS and then you search the databases. In this, we are getting a link known as pathogenesis of AIDS, and this article actually has much information about this HIV, and it is also having information about the drugs used against the HIV infection. So in this page will contain the drugs against the HIV proteases, and there are about 4 drugs are listed here. And these 4 drugs are actually selected for our present project work as our drugs. So these 4 drugs are selected and they are all listed here as a seconavir, ritonavir, ritonavir and melphalanavir. So these are the 4 drugs are selected for the present study. So we identify target, validated target, and we get the drugs for against the target by using this journal. And in order to carry out further steps of drug designing, we need the SMILE forms of these drugs. So we have to search either chemical databases or drug banks for getting the smiles of these drugs. For example, I want to get the smile forms of this particular drug, then I should go to drug bank. As usual, you can go through the Google search, you can go to the drug bank website, and after reaching the website you type the name of the drug and you click the search button, and it will give the link of the drug if it is available. If it is not available here, you should go to a chemical database. So fortunately the drug bank contains this particular drug, and then you should collect the smile format of the drugs by this link, and then PDB file of the drug is also collected by using this link. So like this we can collect the PDB as well as the smile format of the drugs, and then you will combine all the smiles of the drugs into a single file as known as drugs.smile file, and then each PDB file of the drugs is also saved as such. So now we completed the third part of the drug design, that is we identified a target and we validated the target, and now we selected the drugs. Okay, now how you will get the hits? This is the part of the QSR studies. In the previous tutorial itself we explained how we can use the QSAR studies to collect the hits from a chemical library. So by screening the chemical library through this virtual screening method, by using the technique of QSAR, we can identify the hits. So now by using that QSAR studies output, we will use the hits here, because in that QSAR study also we used this drugs alone. Okay, so we can use the hits of that QSAR studies in this method. And then docking, it is also explained in the previous tutorial, that is we use the same enzyme SIV proteases as well as the drugs and hits obtained from the QSAR studies for docking purposes. So now we have all the docked complexes as well as the drugs. For the docked complex it will have a score, as well as for hits we have a score, and the scores are also listed in a separate file. You can see a file is known as drug store. Drug score will be there. So after carrying out the docking, as indicated in the previous lectures, the scores of the first hit of the docked complexes are noted here for 4 drugs as well as for the 3 hits noted here. Then so we completed the target identification, target validation, selection of drugs, selection of hits, and docking is completed. So then we need the other step has to be carried out as ADME and toxicity prediction. The word ADME means is nothing but absorption, distribution, metabolism, and excretion. Then toxicity means whether the drug what we are going to use is going to be toxic to the organism. So that is known as the toxicity prediction. So we are going to predict the value of ADME and toxicity for both drugs and hits, so that we can compare which hit is the better hit. So for the prediction of toxicity we can use some softwares, or rather we can use some online tools. So here we can see the online tools like free ADME is available, and then Mall Inspiration tool is available. So these two tools actually used to predict the ADME values and toxicity values. So how we are going to reach this tool? You can see now this Mall Inspiration tool. So you just go to the Google page and then you type Mall Inspiration. Okay, Mall Inspiration server is there, and then the Mall Inspiration server you can see this calculation of molecular property prediction, and then this is the page which arises. And here there are 2 options available. You can either draw the molecule or you can enter the smile format. So we already have the smile formats of the drugs with us. Say for example, I will copy one of the smile format of the drugs and I can load the drug smile format here, and then I should click the calculate properties option. Okay, now the drug structure is loaded here, and then it has predicted important properties of the drugs here, especially the log P values, TPSA values, number of atoms, molecular weight, hydrogen bond acceptor, hydrogen bond donor, and number of violation. This number of violation means that it is the outcome of a Lipinski rule actually. So that is what the Y number of violation. So I will explain a little bit later what do you mean by this Lipinski rule and violation and all. So we have to keep the value of this log P and this molecular weight and number of hydrogen bond acceptors and hydrogen bond donors into a separate file. Here you can see as a log P, molecular weight, hydrogen bond acceptor, hydrogen donor, and number of violation to a separate file. So like this the values are noted for each drug as well as for each hits. Okay, so you can even the bioproductivity activity of the drugs can also be predicted by using the mole inspiration server. So this is how you can predict the bioproduct activity as well as the chemical properties of the drugs by using this mole inspiration tool. And then you can also save this file by using this file save options. So we predicted by using this mole inspiration server. Then one more option I had mentioned there is pre-admit tool. That is the other one we mentioned there. That is the pre-admit tool, and this tool can also be used for predicting the. It is also used for predicting the ADME, that is especially toxicity. ADME tool, it is used. You can see here, and these two tools mainly used to predict the toxicity of the drugs or the chemicals. So this tool is actually needs a login option. So you have to get prediction of this tool. You have to first log in. Then only you can get the prediction. So here we use this tool to predict toxicity. So for this, again you need the mol format file for the prediction of toxicity. So we should not worry about the format what it requires. You simply have your drugs in the PDB format. Okay, so you just open this PDB in a Marvin sketch application. So once the application is loaded, you can simply save the file in a mol format. See this. So now we can save the file in mol format. So now if you come to this folder, there is a mol file is there. Now open the mol file. By using the source, and then by using this edit copy option, and then you paste this, and then you click load, and now the file is loaded. And once the file is loaded, you just click the submit button, and then it will give the prediction of toxicity from this pre-ADME tool. See now this actually ADME tool identified Ames test toxicity to the chemicals, and then whether it's a carcinogenic activity of the chemical, that is drug against mouse or rat. They analyzed all those things. If it is giving a negative means it is not causing any carcinogenic activity. If it gives a positive means they are going to affect it. And then here also the mutagenicity of the drugs are analyzed. Only it shows a mutagenicity against this important strain of in this TA strain. Only it shows a positive effect. Otherwise for other strains the mutagenicity is not identified. So these are the microorganisms against which the mutagenicity is tested for different Salmonella strains. and then Ames test is also going to be carried out. And since you are getting one output of positive, it gives that the chemical might be like mutagen. So that is what the toxicity for this drug is, shows that. And similarly, you can predict the toxicity for other drugs also, as well as hits, by using this admit. So part of drug designing, we did this ADME toxicity prediction also. For drugs like this, we can predict the toxicity as well as ADME values for the drugs as well as hits. After predicting these ADME values as well as the drug scoring, by comparing the scoring of the drugs, you can see this. When you compare the score of the drugs, the one drug will have a maximum score, that is 8396, and one of the hits which having a maximum score is hit 1. Okay, so out of these 3 hits, the hit 1 is found to be comparable with the drugs. Similarly, when you come to the drugs as well as hits of this ADME as well as the toxicity prediction, you can see this. The drugs will have violation around 3, 4, 2 rule, 4 rule, 3 rule. The minimum is 2 rule violation. All the 3 hits will have only 2 rule violation. So they are almost better than the other drugs compared to the ADME values. So you can select any one of the hit, but comparing this docking score, it's better to select hit as our best lead out of these 3 leads or hits obtained from this QSR study. So by comparing this ADME values as well as the docking score, we are selecting hit 1 as a good lead for further in vitro and the clinical studies to confirm it's a best drug against the proteases. And before this, why I selected this hit by using this violation code? What do you mean by this violation? So this part comes to the rule known as Lipinski rule. Actually, Lipinski rule states that an orally active drug has no more than one violation of the following criteria. Unfortunately, whatever the drugs we are selected, we have violations more than 3. But actually, the Lipinski rule states that we should not have more than one violation. But already we have more drugs will have more violations. So the hit is going to be anyway better than the already available drugs like this 220, 503 and 232. They have more than 2 violations, whereas our hit will have only 2 violations. So it's how it's going to be a better one, but it is not best, because Lipinski rule says that you should not have more than 1 violation. So what violation means? The drug should not have more than 5 hydrogen bond. The drug should not have more than 5 hydrogen bond donors. Okay, and then we should not have 10 hydrogen bond acceptors. So here hydrogen bond should not be a bond alone, it should be a bond donors. Then it should not have more than 10 hydrogen bond acceptors, and then molecular mass should not more than it should not be more than 500 Daltons, and then its log p value should not be greater than 5. Why it is called a rule of 5? Because all these values comes as a multiples of 5. You can see 5 hydrogen bond donors and 10 hydrogen bond acceptors and 500 Daltons, 5. So these are all comes under the multiples of 5. So the rule is known as Lipinski rule of 5. But the rule number of rules is only 4: hydrogen bond donor, hydrogen bond acceptor, molecular weight, and log p values. So you predict the ADME values and then compare the ADME values with this Lipinski rule. On the basis of this Lipinski rule, we are selecting best lead. So that is how we selected the hit 1 as a best hit compared to the other hit, and it is considered to be as a drug likeliness compound. So if any drugs which obeys this Lipinski rule, then it is called as a drug likeliness compound. So that's what you can see here. It's a best lead and drug like molecule. After this, this cannot be marketed directly. You need to carry out some preclinical wet lab studies as well as clinical trials. So after this preclinical as well as clinical trials, we can convert this drug into a we can convert this lead into a drug, and then it will come to the market. Okay, so this is how we predicted a drug for protease. Thank you.